Welcome to this lecture on fleas as insect vectors. When we think of fleas, we often picture a frustrating nuisance for our pets. However, in the grand scheme of nature and human history, these tiny, wingless insects are far more than just an annoyance. They act as biological syringes, carrying dangerous microscopic pathogens that have literally altered the course of human history. Today, we will build a complete understanding of how they operate, how they transmit diseases like the bubonic plague, and how public health officials use mathematics to predict and prevent deadly outbreaks. To begin our journey, we must first understand what makes the flea such a formidable vector. Imagine a tiny ninja, no larger than a sesame seed, possessing the superpower to jump over 100 times its own height. This incredible mobility allows them to easily hitch rides on rats and jump onto humans. But their true danger lies in their role as biological syringes. They move from one host to another, carrying dangerous microscopic pathogens with them. By doing so, they have been responsible for some of the most devastating zoonotic diseases in human history, including the infamous Black Death. Understanding their basic operation is the first step in realizing why such a small insect commands immense respect in the medical world. Now that we appreciate their historical impact, let us look at their biological classification. In medical entomology, fleas belong to the insect order Siphonoptera. This name translates to wingless tubes, perfectly describing their lack of wings and their specialized tube-like mouthparts designed for piercing skin. They are classified as obligate hemotophagous ectoparasites. Breaking that down, it means they absolutely must feed on the blood of warm-blooded hosts, and they do so from the outside of the host's body. Unlike mosquitoes that fly to find their next meal, fleas rely on their powerful hind legs. These legs are powered by a highly elastic protein called resilin, which acts like a coiled spring, allowing them to leap onto passing mammals or birds with astonishing speed. Understanding their physical traits leads us to the actual mechanism of disease transmission. Fleas do not just mechanically carry pathogens on the outside of their bodies. Instead, they act as biological vectors. This means that when a flea ingests infected blood, the pathogen, such as the plague bacteria, actually multiplies and develops inside the flea's gut. As these bacteria multiply, they form a massive clump that physically blocks the flea's digestive tract. The flea becomes starving and aggressively tries to feed again. When this blocked flea bites a new host, the blood cannot pass the blockage. As a result, the flea regurgitates the multiplied pathogens directly back into the fresh bite wound, effectively transmitting the disease to the new host. Let us visualize this transmission mechanism with a clear diagram. On the left side of the screen, you see the rat reservoir, which is the original source of the infection. The flea feeds on this infected rat. Following the arrow to the center, we see a magnified cross-section of the flea vector. Notice the dark mass inside its digestive tract? This represents the blockage caused by the multiplying plague bacteria, Yersinia pestis. Because of this blockage, the flea cannot digest new blood. Finally, look at the right side of the diagram. When the starving flea attempts to feed on a human host, it regurgitates the bacteria directly into the bloodstream. This visual perfectly captures the biological bridge the flea creates between rodents and humans. Having seen how transmission occurs, we must now consider how we protect ourselves. The study of fleas is critically important because they transmit severe zoonotic diseases like the bubonic plague and murine typhus. If we did not deeply understand the relationship between rats, fleas, and humans, we would be completely defenseless against sudden outbreaks. In modern public health, epidemiology teams do not just wait for people to get sick. Instead, vector control teams actively monitor flea populations in both urban and rural environments. By keeping a close eye on how many fleas are living on local rodent populations, scientists can issue early warnings. This proactive monitoring allows them to implement targeted pest control measures before an epidemic starts, ultimately saving countless lives. To actively monitor these populations, public health officials need a precise mathematical tool, which brings us to the specific flea index. Conceptually, without a numerical way to measure flea populations, we would just be guessing whether a city is safe or in danger. This formula translates biological field data into a clear, actionable value. The formula is I equals F divided by R. Here, I represents the specific flea index, which is simply the average number of a specific species of flea per host. The variable F stands for the total number of fleas collected during a field survey. The variable R represents the total number of hosts, usually rats, that were examined. If the total number of fleas increases while the rat population stays the same, the index will increase, indicating a much higher risk of disease transmission. Now that we have our formula, let us apply it to a real-world scenario. Imagine a public health team in a coastal city tasked with monitoring the risk of bubonic plague. They set up traps and successfully capture 50 black rats. Upon careful microscopic inspection, they collect a total of 85 oriental rat fleas from the fur of these rats. We are asked to calculate the specific flea index. We start by identifying our given values. The total number of rats, R, is 50, and the total number of fleas, F, is 85. We choose the specific flea index formula because it directly calculates the average vector load per host. We substitute 85 for F and 50 for R. Dividing 85 by 50 gives us our final answer of 1.7 fleas per rat. This number represents the average density of the vector population. We have calculated an index of 1.7, but what does this number actually mean for the city? In epidemiology, a specific flea index greater than 1.0 is generally considered a critical warning sign for a potential plague outbreak. Because our calculated value of 1.7 is significantly higher than this safety threshold, it physically means that the flea population is dangerously dense. This high density allows the plague bacteria to easily sustain itself and rapidly spread among the rat population, eventually spilling over to humans. Consequently, the public health team cannot ignore this result. They must immediately initiate flea control measures, such as dusting rodent burrows with insecticides, to bring the index down before attempting to reduce the rat population itself. This brings us to another crucial aspect of vector control: environmental changes. Environmental factors play a massive role in the life cycle of insect vectors. Let us look at a second example to evaluate this. During the hot, dry season, a surveillance team examines 120 rats and finds a total of 90 fleas. We need to calculate the flea index for this season. We use our standard formula, substituting 90 for the total fleas, F, and 120 for the total rats, R. Dividing 90 by 120 gives us a final answer of 0.75 fleas per rat. Physically, because this number is below the critical threshold of 1.0, it indicates a relatively low risk of disease transmission. The lack of environmental moisture likely prevents flea larvae from surviving. Now, let us see what happens when the weather changes. 6 months later, the exact same team conducts a survey during the highly humid monsoon season. This time, they examine 80 rats and find a massive total of 200 fleas. We again apply the flea index formula to determine the new vector density. We substitute 200 for our flea count, F, and 80 for our rat count, R. Dividing 200 by 80 yields a final answer of 2.5 fleas per rat. This number is extremely high and sits well above the established danger threshold of 1.0. The physical meaning here is clear. The humid conditions provided a perfect incubator for flea eggs and larvae to thrive, resulting in a dangerous population explosion. Let us compare the results from our two seasonal calculations. The calculated flea index jumped dramatically from 0.75 in the dry season to 2.5 in the monsoon season. This stark contrast physically demonstrates how higher humidity and warmer temperatures create optimal breeding conditions for fleas. It is not just a slight increase, it is a massive population explosion. For public health officials, this data is a clear call to action. They must be especially vigilant and proactive with vector control right before the monsoon season begins to prevent this dangerous spike. If they wait to act until the index already reaches 2.5, an outbreak of flea-borne disease might already be underway in the human population. We have covered a lot of ground today, so let us review the most important ideas. First, we learned that fleas are highly specialized biological vectors. They do not just carry diseases, they incubate them and transmit them by regurgitating pathogens into bite wounds. Second, we explored the specific flea index, a vital mathematical tool that allows epidemiologists to quantify the risk of an outbreak by calculating the average number of fleas per host. Remember that an index above 1.0 is a critical warning sign that demands immediate action. Finally, we saw how environmental factors, especially humidity, can cause vector populations to explode. By mastering these dynamics, we empower ourselves to protect vulnerable human populations from these ancient but persistent biological threats. Great job following along with these concepts.